Week 1, we did isolation and concentration of DNA. The DNA matter was derived from a fecal sample from each specimen through isolation using the Qiagen spin column and the QIA-AMP DNA stool kit, according to the manufacturer's guidelines. The purpose of the kit was to isolate the DNA from RNA, proteins, and other sources of contamination. DNA concentration. The extracted DNA was then concentrated to increase the amount of DNA present in the sample using the Zymo kit. Zymo spin column concentrated DNA by 10 times. This was conducted by James Stegman according to the manufacturer's guidelines. Week 2, we did quantification of DNA samples in PCR. We used the UV spectrophotometer to determine the concentration of DNA if there was contamination. DNA absorbs light at 260 nanometers, and proteins absorb light at 280 nanomolars. So we used the 260 nanomolars over 280 nanomolars ratio to test our purity. Amplicon PCR amplifies the DNA, specifically the V3 and V4 regions of the 16S ribosomal RNA. We did denaturing, annealing, extension, and then 30 cycles over 1 billion copies. Week 3, we did gel electrophoresis, amplicon PCR cleanup, and indexing PCR. We used a gel electrophoresis. A gel electrophoresis analysis was conducted using an Aerorose gel to determine if the PCR was conducted successfully. It was conducted using a Lonza flash gel system charged at 250 V for 5 minutes. Amplicon PCR cleanup. Using the Agincourt Ampure XP kit, the samples were cleaned to remove any unused materials from PCR such as primers, DNTPs, enzymes, and other impurities. Indexing PCR. This was performed to place tags on the samples for identification purposes when sequencing using the Promega Corporation kit. We did 95 degrees Celsius for 3 minutes, 8 cycles, 95 degrees Celsius for 30 seconds, 55 degrees Celsius for 30 seconds, and 72 degrees Celsius for 30 seconds. Week 4, we indexed PCR cleanup, fluorometric quantification, and dilution. Index PCR cleanup. Using the Agincourt Ampure Kit XP kit, the samples were cleaned to remove any unused materials from PCR such as primers, DNTPs, enzymes, and other impurities. Done once again to ensure that all the impurities are washed away. The fluorometric quantification. Using the fluorometer, the index samples were quantitated to determine if the indexing PCR was successful. The dilution. Calculations were performed to determine the volume of water needed to dilute the samples to make them 4 nanomolars in concentration. Week 5 was DNA sequencing. Illumina MISI Next Generation DNA Analyzer determines the nucleotide sequence of each sample. Indexing PCR products are denatured, then bind to a chip. Each DNA molecule is then amplified. So our results, the samples that we used, sample A was number 332. The amount of DNA per sample was 100. Concentration was 158.1. And purity using 260 over 280 ratio was 0.63. Our sample number 442 had 100 for the amount of DNA per sample. Concentration was 44.6, and the purity using 260 over 280 ratio was 2.24. Number 361 was 100, the same as the rest for the amount of DNA per sample, and the concentration was 73.5, and the purity using 260 over 280 ratio was 1.36. Our sample number 430 had 100, the same as the rest. The concentration was 98.5, and the purity using the 260 over 280 ratio was 1.01. This is the gel electrophoresis results. These were our results for all 4. The only two that ended up working was number 361 and our negative control. Or our positive control worked, our negative control did not work. Sample number 332 had nothing on the fluorometric reading. 442 had nothing on the fluorometric reading. 361 had 1.46 on the fluorometric reading, and 430 had nothing. Our positive control had something, but it was not enough to be recorded, and our negative control had nothing. Our calculations of the dilution ended up coming out at 4.015 nanomolars. No further calculations were made as the DNA sample was concentrated enough. So this is just the total number of reads for each sample sequenced. So we had for 361A, we had 8,128 reads, and then the percent of reads past quality filtering was 100. Our other sample, 361, had 28,296 reads, and the percent of the quality filtering was 100. Our positive control had 44 reads, and the percent of reads passing quality filtering was 100. These are our two figures of the kingdom classification for our samples. So the first one here is our first sample, 361A, and the second one over here is 361B. So the bacteria is 8,104. The unclassified at kingdom level was 18 for the number of reads. And on the other one, the number of reads for unclassified at kingdom level was 62. And the bacteria was 28,225. This is our figure that shows the sample classification by kingdom. So A is the blue and B is the red for bacteria. Our top phylum classification results for sample A, which is over here, shows that there is less of a significant difference than on this one over here. That's about it. This shows our sample classification by phylum. So the Firmicutes has a bigger difference than the bigger difference for sample B than sample A, and the same with the rest of them, the bacterioids, the Protobacteria, unclassified, and Verocomicron. Top order classification results shows that again, sample B has a lot more than sample A. Sample classification by phylum, here's just another showing that B is a lot bigger than A. The top kingdom classification results for the positive control sample. So bacteria was 44 reads, it was 100%, and there was nothing unclassified at kingdom level. The top phylum classification results, so this just shows that 31.82% of Firmicutes showed, the 20.45% was Proteobacteria, other was 4.55, and the Bacteroidetes were 43.18. Here's some more results on the top classification results for the positive control sample. Our discussion was, though our experiment was adequately designed to test our original hypothesis, it was not able to be tested due to factors like sample degradation over time. We were forced to change our hypothesis to test a new theory. In future experiments to adequately test our hypothesis, we will need to verify how fresh our samples are and also add in multiple tests of each sample to ensure that we get the most accurate results, and can see if any parts of the samples provided might have more usable DNA than other parts. When we went through UV spectrophotometry, all of our samples had absorbances well above 6.7 nanograms microliters. This could have been due to contamination of RNA or proteins that were left over and had gotten trapped in the filter with the DNA. This could also have resulted in the purity readings being within acceptable limits of 1.6 to 1.8 for our samples. When we did electrophoresis, neither of our samples had any DNA in them, but our secondary sample had one reading, number 361, at the 550 BP range, along with our positive control, which means that no DNA was present in the other samples, or our negative control. This means that all of our other samples were contaminated with either proteins, RNA, or both. And that is all.